Here I want to expand on the restriction enzymes. I have drawn out two commonly used restriction endonucleases used in a molecular biology lab. One is called EcoR1, and it recognizes the sequence GAATTC. So remember, it's palindromic. So here's what it looks like in a double-stranded form. So you can see the GAATTC in this 5' to 3', and then GAATTC in this 5' to 3'. If I cut it, it leaves these types of sticky ends. So essentially what we're saying here is for this to be used to kind of glue back together, I need to have an exact match here. That means on another strand here, I need to have an AATT. Here I've shown another restriction enzyme called BAMH1. It recognizes GGATCC, and it cuts right here, and it leaves these type of sticky ends. So what I want to demonstrate to you is if I try to glue one piece that was cut with EcoR1 and glue it together with another piece here that was cut with BAMH1, this is how they would match up. So G to C, G to T, that's not a match, so those are not going to stick together. A to T, we got complementary base pairs here, that's good. T to A, complementary base pairs here, that's good. And then C to A. Now, those aren't going to match. So we don't have a 100% match here. So these are not going to stick together. We need to have a 100% match of these complementary base pairs for these sticky ends to stick. So what this means is we can't use these two together to create a bigger complex. These two restriction enzymes, if I cut two different pieces with them and try to glue them together, because the complementary base pairs do not match 100%, they will not be able to glue together. But if we go back over here and we cut with EcoR1 and then try to glue it back together, it will indeed religate together. And what I mean by this is if I put it under the right conditions, these AATT will match up with this complementary TTAA. So then all I need to do is reestablish the phosphodiester bond that will exist between the 3' and the 5'. So remember, on the 5' is the phosphate group, and on the 3' is the OH group, or the hydroxyl group. So because the complementary base pair will stick them together, so if I draw it, it's going to look like this. So here we have it. The complementaries stick back together, but what I need to do is re-glue these joints together. And what we use is an enzyme, and that enzyme is called ligase. So ligase is involved in reestablishing the phosphodiester bond and essentially gluing them together. If we did not add the ligase and do that reaction, if you just heat it around or moved it too much, these complementary base pairs or weak hydrogen bonds, they'll pop apart. So what we're doing is we're gluing it together with a nice covalent bond on the same strand, and this glues those together. So typically when we do an experiment in the laboratory, we're going to cut with restriction enzymes that are compatible with each other so they match up, and then we'll subsequently treat it with ligase so we can glue the matching parts together. So another way to schematically draw that is to put it like this. Here I have my gene, if you remember, I was interested in. So I can cut that with EcoR1. I'm just going to abbreviate R1. I am then going to have my plasmid. And in my multiple cloning site, hopefully there's an R1 site or an EcoR1 site. So I'm going to cut here, cut here, cut here. What this is going to do is give me this fragment with R1 sites on either side. And when I cut this, I linearize it. Or a better way of drawing it is I kind of pop it open. And on each site is an R1. So let's ignore this. But if I do cut a circular piece of DNA once with a restriction enzyme, I create a linear piece. But here I pop this vector open with my restriction enzyme. I pop my piece out with my restriction enzyme. These are the same, so they're compatible. So I can put them in, do what we call that ligase reaction, what we call ligation. And then what I create here is my plasmid with R1 sites on either side and my gene A in it. Now, since this is in a vector, I can now insert that into bacteria and copy it, and so I can make multiple copies of my gene A.